Hi, to everyone in the room. We have a lovely full room, which you guys online can't see unless I pick up the computer, and I'll probably drop it if I do that. So we're not going to do that. But we have a lovely audience here at Gump Station in Moorea, and welcome to all of you online as well. And thank you for joining us for our culminating symposium for this amazing journey that we did for marine biodiversity and conservation, which was S321 with the Education Association. And our students are really excited to share with you guys today. So I wanted to start by saying, usually when I start a talk, I do a land acknowledgement slide to acknowledge that we are all on native land. But I like to think of it in this context as a sea acknowledgement, especially since the sea is so important to Polynesian cultures, that we started this journey in Aotearoa, New Zealand, and we ended here in the Society Islands on Moorea and Tahiti. And so we, as a group, would like to acknowledge the amazingness of the culture and the people that have shared so much with us. And our students themselves wrote an acknowledgement, so I also want to share that with you. Wow. Okay. Hopefully. Yes. As sailors and scientists, we would like to thank those that played a crucial role in obtaining and synthesizing our research. Thank you to the S321 students and crew aboard the SSV Robert C. Siemens and the UC Berkeley Gump Station. Immersed in and studying the natural and cultural elements of the South Pacific Ocean has been an immense joy. With this research, we hope to inspire others to care for, protect, and advocate for the South Pacific Ocean and those who inhabit the Pacific Islands. We would like to acknowledge the communities, cultures, and indigenous presence on these islands, and it is our hope that these voices continue to be uplifted and heard. So I just want to say how proud I am of all of these students. We had an amazing and at times very challenging journey. I'm sure they're going to come home and tell all of you family and friends about that. And we encountered some pretty amazing sea states and winds. But they also grew as people throughout this journey. And it was amazing to watch that. And it was so fun and rewarding to be a part of that. So thank you all for making this what it is. And I'm so excited that we get to share with so many people that we care about this research that you've done and this journey that you've had. So with that, I am going to try to switch slides on this computer here and bring up our first presentation, which isn't scheduled to start until 9:30, but it may actually take us that long to figure this out if this computer is still this slow. So hang in there. I'm going to mute. Okay, so we're going to welcome our first group is going to talk to you about the Portuguese man-of-war Physalia and what we encountered and some interesting questions in the South Pacific Gyre.
Wonderful. Thank you all for being here. Thank you, Sarah, for the lovely intro. I'm Anna, this is Robin, that's Gabby, and that's Amy. And as Sarah said, we're talking about the Portuguese man-of-war, otherwise known as the blue bottle, or we'll be calling it by its scientific name, Physalia, and its species distribution in the South Pacific Gyre.
All right, so if you don't know what a Physalia is, it looks like this. It's not actually a jellyfish, even though it looks like one. It's a siphonophore, which are related to jellyfish, but they're colonial predators that live in the ocean. And if you've ever seen a Physalia, you've most likely seen it like this, washed up on a beach, either dead or dying. And a lot of science has been done on Physalia that have been washed up on beaches because they're easy to access. But we wanted to study Physalia in the open ocean, which is their natural habitat. And so in the open ocean, they look something more like this, which is one that we caught in a bucket, and we put it in a bucket. And Physalia have a float, which helps them stay on top of the water, and then they also have a sail, which is that part on the side. And normally it's sticking up and it catches the wind and it helps them move along the surface of the water. And then they also have tentacles, which is how they catch their food. And it's being really slow, sorry. Okay. So previously, scientists thought that a lot of open ocean species were really well genetically mixed, and there weren't a lot of barriers to this genetic mixing. For example, they thought that copepods and ctenophores, or comb jellies, were really well mixed. There weren't barriers to their genetic mixing, and they were all kind of really similar. But they recently found out that there actually are barriers in the ocean that are harder to see because it's the ocean and it's not land. There's not mountains that are preventing this genetic mixing from happening, but they found out that there actually are barriers and there are genetic differences between copepods and ctenophores. And they found that what's driving this differentiation in copepods and ctenophores is a large part due to currents.
Yeah, so Physalia are another group of animals that people thought had a pretty worldwide genetic distribution. They thought that they intermixed all together, but that is actually not true. The Physalia in the ocean look a little bit more like this. There are more than one species. Whoa, crazy. People have recently been doing this work. It's been going on a little bit since the 90s, but the main people that are working on this project are from the Dunn Lab at Yale. It's headed by Sam Church, and they figured out that there are at least 4 species of Physalia. There's either 4 or 5. And they figured this out by using a little bit of citizen science and then some genetic analyses. So they had people send in pictures of Physalia that washed up on beaches, and then they morphologically ID'd them and sorted them into what they thought were the species. And then they ran genetic analyses also on these Physalia that washed up on beaches to figure out that there are multiple species of Physalia. And like copepods and ctenophores, people didn't really know what distributed them for a while. And copepods and ctenophores are mostly moved by current, but they're also influenced by things like temperature and salinity. But copepods and ctenophores don't have a sail. Physalia do have a sail. Whoa, crazy. So we thought that maybe wind would be influencing their distribution.
Yeah, so we hypothesized that wind would influence the distribution of Physalia that we found along our cruise track. And then we also hypothesized that along our cruise track, in the different regions that had different prevailing winds, we would see different lineages of Physalia.
So this here is what sampling looked like over the Siemens. This is how we collected our Physalia. It's called a Neustron net. It skims the surface of the water, and we towed this net mornings and evenings twice daily as weather permitted, which it certainly didn't always do. And here's a map of all the stations that we conducted. Out of 48 stations in total, we encountered Physalia in 16 of them. And you might notice this trend here that once we entered the tropics, we stopped encountering any Physalia. And in total, between all 48 stations, we collected 79. And we were interested in comparing them to environmental data. The Siemens is equipped to continuously monitor different factors like sea surface temperature, salinity, current, and importantly, wind. This is a map of all the wind that we encountered, direction and magnitude over the course of our journey. And in order to analyze it, we divided this cruise track into 3 general geographic regions. The first over the Chatham Rise, the second along the southern edge of the gyre, and the third towards the center of the gyre as we traveled north into the tropics. And we found that in these 3 regions, there were distinct prevailing wind directions. So over the Chatham Rise, the wind was coming from the west, over the southern edge of the gyre from south-southwest, and in the center of the gyre, east-southeast.
So after we collected our Physalia, we needed to extract their DNA so we could do sequencing on them. And so in order to do that, we removed the tentacles from 28 Physalia. And then with that DNA that we extracted, we did whole genome sequencing, which means we looked at the entire genetic makeup of each of the individuals for 28 Physalia. And then after that, we needed to look for a special gene called the CO1 gene, which helps us identify species. And so we were able to find 17 CO1 genes out of the 28 individuals that we sequenced. And because we only got 17 CO1 genes, we also combined our data with the data from last year's similar cruise track with their CO1 genes that they found.
So then we wanted to turn all of that sequencing data into relationships in between our individuals. The way we do that, you can imagine if we only looked at one spot in the genome, we looked at one AC tier G, we could put that on a 1-axis chart. If we looked at 2 spots, we could put it on a 2-axis chart. If we looked at 3, we could get to 3-axis, but we did this with 84,000 different spots in the genome, which is hard to put on a chart. So what you do is you do some analysis to turn those 84,000 axes back into 2. which then get these strange names, but they do explain about 75% of the variation. And all we're looking at is things that are far apart are genetically distant from each other. Points that are close together are genetically similar. So of our 28 sequenced Physalia, 25 formed a pretty tight cluster that looks kind of like a green blob over there. And then 3 were very distant from that group. 2 of them were found towards the center of the gyre, and those were the only 2 that we found towards the center of the gyre. One of them is a little strange. It was found towards the beginning of our cruise track along the Chatham Rise, and is much closer to those central gyre animals than it was to that big cluster. Now, one of the issues with this chart is we can't see what's going on in that cluster because we're so zoomed out to get all of the data. So when we zoom in, we can see that all of our animals, or most of our animals from the Chatham Rise and the southern part of the gyre, are forming this well-mixed population. We don't see a distinct green group and a red group, so that tells us that this is one species that's well-mixed. And we are able to identify, with some help from the Dunn Lab and from last year's data, that this represents 2 different species that were identified in that paper. So our tight cluster that we found farther south and on the Chatham Rise is an unnamed Physalia species that we'll just go by Physalia species. And then our distant from the group animals were Physalia megalista. Then we wanted to take a look at those CO1 genes that help us identify species and tell differences in between them. And so we made a CO1 map. This is kind of a weird looking figure, but all it is that each circle represents a number of individuals that have the exact same genetic sequence in this gene. So it's a short gene, and each tick mark is a 1 base pair difference. So in an entire genome, a 1 base pair difference would be nothing. But because this is such a short segment of DNA, a 1 base pair difference is somewhat significant, and a 4 base pair difference is more significant. So what this tells us is that that big group, if green and red, which is all of the animals that were found in the Chatham Rise and the southern South Pacific Gyre, represent Physalia species. And there's some differences within there. They get as far as 2 base pairs distant from each other, but ultimately it's one population with some things going on. And then this other group that's 4 base pairs away represents Physalia megalista, and those were the animals that even last year were only found in the central South Pacific Gyre.
So as a result of our genetic analyses, we were able to conclude that these regions with distinct prevailing wind directions had distinct Physalia lineages present. So as you remember, the first 2 regions was where we found that Physalia species, and the center of the gyre was where we found Physalia megalista. And of course, there are other factors that we would expect to vary geographically. Sea surface temperature, salinity, current could all play roles in influencing distribution. But for this sailing neustonic species, wind does appear to be a contributing factor.
Yeah, so why do we care? This is all cool and all. There's 2 different species of Physalia in the ocean. Cool. Well, the information done on this Physalia species is not really done anywhere else. There's not a lot of sampling that happens in the South Pacific Gyre because it's kind of hard to get there. We were in the middle of nowhere. Also, this information for our Physalia speciation could also add to provide some hints on what causes the speciation of other zooplanktonic species. And lastly, the genetic data that we collected here is added to the UN's biodiversity beyond national jurisdiction goals of creating a whole open ocean genome. And that genome and the information will then be used to share scientific knowledge with all of humanity, basically, and try to encourage more conservation in the open ocean.
With that, thank you. And are there any questions?
We can start with questions in the room, and then Kayla can monitor our online audience.
What were some challenges you guys had, that you guys experienced with your data set as you're going on our cruise track?
Will you repeat the question for the microphone too? Because I feel like
The question was, what were some challenges we experienced with our data set as we moved along the cruise track? So we had a small sample size in the central South Pacific Gyre, and partly we were able to mitigate that with last year's data, but this is one of the powers of being able to do repeated sampling where we do this cruise track year after year, is that we can supplement those things. But this year, it was really a small sample size in the middle of the gyre that it was very different. And so that still tells us something interesting, but it would be great to have more data there.
We also had a gale when we were in the central South Pacific Gyre, which prevented us from doing new stontos and collecting Physalia, so that also probably was part of the reason why we had such a small sample size from that area.
So potentially related to challenges, Subek asks, do they sting?
They do indeed. Yeah.
Like so backwards star.
Yeah, all of us were able to make it through this whole journey with no stings. I don't think anybody in the ship's company was stung by.
Yeah.
Well,
One person was stung by Physalia.
That's not the culprit there.
No, we don't think it was like
Yeah, but we did take a lot of care in handling them carefully when we were doing our processing and when we were handling the net toads that contained Physalia.
They said that it's related to the movements, right? Human sticks to the prevailing species of beast. So like to the beast. Like from the also
It's hard to say with the data we have.
Repeat the question.
Oh, sorry. The question is that here in the Society Islands, the prevailing winds are coming out of the east. So to that end are the Physalia that are found in this part of the world coming from the east. Maybe. This is an area, especially in the open ocean, where very few studies have been done. The church paper that a lot of our project is sort of built off of, all of their animals were washed up on beaches. So it's hard to know how things are moving across the ocean because it's, well, we found these things in coastal New Zealand and coastal South America, and we don't know what's happening. And so that's part of the value of taking ships out and going and doing science on the open waters. It's hard to say with the data we have right now.
you would have to do a study in between here and the US to figure out if they were still safe.
Next year, let's run.
Let's go.
Is there a relationship with Balala, the Salem, the Seminoles, do any work with those?
Not on this journey.
We didn't do any like analysis of Valella while we were on the cruise track, but we did read some papers that said Valella is influenced by certain prevailing wind directions, just like Physalia is.
How they relate?
Like genetically, they're both siphonophores.
They're both siphonophores. They're both,
Yeah.
and they're the two cnidarians that sail. So they're related in that way.
Somehow, yeah.
what are the predators?
Yeah, the question
So
was,
the
what are the
predators.
predators for Physalia?
Turtles. Just kidding. I'm just kidding. They're not turtles.
I'll happily talk about this. So they're these nudibranchs. They're these sea slugs that
knew that.
live in open water. They never hit the bottom. That are also the same kind of bluish color, and they'll munch on Physalia. We found, I think, one floating around across our entire cruise track. But they're also like top of the water little sea slugs that are small and very pretty. Called Glaucus. That's what it is.
I know something.
Are there any more in the chat, Kayla?
Yeah, there's quite a few.
We have time. I started early. Sorry, you guys.
Oh, you're doing it.
Page says, great job. What other environmental factors do you think may influence their distribution?
Wow. What a good question.
Yeah, well, looking at the air-water interface, we definitely expect that currents would play a large role in the distribution, just like the other neustonic species that we researched when we were building up our hypotheses. Our data set was pretty limited as far as like being able to really draw any strong correlations between these factors and the distribution that we saw. So that would definitely be something that like further research and more data collection would help. But yeah, also different water masses that we would encounter, different salinities and different temperatures. We would expect that different species would thrive in different conditions.
It's hard to draw causal relationships in this kind of setting because it's in the same place that we find warm waters, we find different currents. So it's hard to be like, well, it's the warm water of the current that's causing that.
Aaron Bryant asks and says, fabulous. What morphological differences are there with the species preferring different wind directions?
How do I go back to slides?
Well, there it is.
Whoa.
Wait,
Oh,
yeah.
okay.
there you go.
Too many.
It's a little. Ta-da.
So go for it.
So there's a difference in lots of things. Most of Physalia like look pretty similar, hence why people thought they were the same species. But Megalista has this really long float that goes off to one side. Physalia also differ in handedness, which is which way their tentacles come down on their body. So you can kind of tell differences between that. And then if you look very carefully, you can tell that Physalia are different species.
There's our first slide of Physalia bucket. This is not the fastest way to get there, but it's gonna work.
It's fine.
So this Physalia you can tell is clearly a Megalista because you can see that it's got this really long float here, whereas Physalia species would be the sail would be the same as the float pretty much.
Yeah, and as we processed, we actually were taking measurements of morphological characteristics that we're sharing with this lab, the Dunn lab, that has this ongoing project. So I'm sure that's something that they'll be analyzing further.
All right, let's do one more, and then I'll talk about how we can answer more at the end.
Okay. Amelia Lang asks, do other jellyfish have similar trends, or do you think this is a trend specific to the mammal?
In that like there are more species than we think.
Or yeah, maybe, and or wind influencing things. I think those are your two major things.
Okay, so I would say that there are more species than we think probably applies to a lot of things in the open ocean. As we were discussing at the beginning, pretty much the more studies that are done on this, the more times we figure out, oh, these things that all look kind of similar are actually not the same across the entire world ocean. In terms of wind, they're a little bit unique in the fact that they sail. So I think the main influence for a lot of other things would be currents, but unexpected things happen all the time in science, and I wouldn't be completely shocked if wind had an influence on things that don't draw such a neat line in between them.
So wind influences currents.
Yeah.
Possible that wind could play a factor.
Great, well, let's thank the Physalia group. And I'll say, if we didn't get to your question for the in-between, we only have a few minutes in between each talk scheduled. We will have a whole question and answer session after the entire symposium. So we can try to revisit those questions that we didn't answer there as well. So stay tuned. All right. All right, so our next group up is studying a really important set of critters, which happen to include mollusks, which I love. But they're going to talk to you about marine calcifiers across our really unique cruise track in the South Pacific Gyre. Go ahead and take it away.
Yeah. Hi, everyone. Welcome to the calcification craze. My name is Patricia. This is Resh, and that's Brigitte. And we've spent the last semester looking into the effects of ocean acidification on marine mesopelagic calcifiers. So you may be asking, what are marine mesopelagic calcifiers? Let's break that down. What are calcifiers? Calcifiers are marine organisms that use calcium carbonate to form their shells or exoskeletons. Some examples include marine snails. So these are our pteropods and our heteropods. And another one that you might be more familiar with are coral. Another group of calcifiers are crustaceans. So think your crabs and your lobsters, as well as this guy was kind of shrimpy guys, a euphausid, also known as a krill, and a copepod. So it's important to note that for crustacean groups, their shells are made largely out of chitin and not calcium carbonate. So some people do not consider them to be real calcifiers. However, calcium carbonate is a component in their shells, so we are including them as a calcifier for this study. Next, what are some non-calcifiers? These are the more gelatinous things like jellyfish, or what the previous group kind of talked about, nudibranchs. These are the slugs of the sea, basically.
This is actually the species that eats the Physalia.
Wow, that's so cool. So next, what is the mesopelagic zone? The mesopelagic zone is what you may have heard being referred to as the ocean twilight zone. This is around 200 to 1,000 meters below the sea surface, right around there. At this zone, little sunlight reaches it and there's low productivity. And because it's so deep, it's really hard to sample down there. And this is why it's an understudied part of the ocean and why we focused on it during our project.
So we've talked about ocean acidification, but how does it work and how is it related to our calcifiers? So as a lot of us know, humans release a lot of atmospheric CO2. Typically, a lot of this CO2 is uptaken by the ocean. As it mixes with water, it forms carbonic acid. But carbonic acid dissolves really quickly in water. It doesn't stay that way. It loses its hydrogen ions, and then the ocean gains hydrogen ions, decreasing the pH and making the ocean more acidic. And counterparts of those hydrogen ions, carbonic acid becomes either bicarbonate or carbonate. But in order to kind of buffer this acid, the ocean puts the hydrogen ions and the carbonate back together to form bicarbonates. Now, this would be okay, except our critters, the calcifiers, use carbonate. Carbonate is bioavailable carbon that our calcifiers use to form their shells or their exoskeletons. So as we add atmospheric CO2 to the water, our critters have less carbonate to use for themselves, and carbonate saturation, or the amount of carbonate ions in the water, goes down.
So why do we care about this? These are just some little critters pretty deep down in the ocean. What do they have to do with the rest of the environment? And also, what do they have to do with us? So in order to visualize this, we're going to be using the pteropod, but this refers to all calcifiers, what I'm about to say. So this guy is really important for biodiversity as well as our food chain. Little fish eat pteropods and other calcifiers, and then big fish, like a tuna, eat the smaller fish, which are important for fisheries as well as for the marine ecosystem as a whole. So without these little calcifier critters to make the bait lower down in the food chain, these bigger animals cannot exist, and a really important niche would not be filled. Also, they're really important for carbon sequestration. So they do this really cool thing called diel vertical migration. They start down here when the sun is up, deep in the ocean, the mesopelagic. Then when the sun goes down, they migrate up to the top, the surface, and up there they eat things which have carbon in them. So then when the sun rises again, they go back down and they transport that carbon below the surface. They poop and they respire, releasing some of that carbon below the surface. And then they also have all of this carbon when they secrete their shells. There's carbon in those shells. So when they die, they then sink. And they sequester all that carbon down into the deep ocean for hundreds of years.
So, based off of this background knowledge, we had some questions we wanted to ask. We wanted to look at carbonate saturation and its effect on marine calcifier biodiversity. So on this cruise check, we had two tools that we were able to use to measure biodiversity. One is relative abundance. So we were looking kind of at each sample, what number of calcifiers versus non-calcifiers were there. And the other biodiversity measures species composition. So we're looking at the presence and absence of like certain species in each sample.
So how did we actually collect these samples? We used two types of nets. We used the meter net, which is on the left, and we used the Newston net, which is on the right in the middle. We deployed these every night. We only used nighttime tows. And the meter net goes below the surface to about 150 meters. Newston net stays on the top, but they're both catching those dial vertically migrating mesopelagic critters. We also used this hydrocast carousel in order to collect our water samples.
So in our questions, we talked about some variables, carbon saturation and biodiversity, but it's important to talk about how we define and measure those for this project. Now, carbonate saturation, as I said before, is the amount of carbonate ions in the water. that is typically measured by aragonite saturation. Aragonite is a type of calcium carbonate. It's the most soluble type. And aragonite saturation measures how many carbonate ions there are in the water relative to their ability to come together, precipitate, and actually form those shells. Now, as we just discussed, our critters undergo vertical migration. So they're exposed to a large range of carbon saturation throughout their day. And it was important that we encapsulated that for this project. So what we did is we took aragonite saturation at depth and subtracted it from aragonite saturation at the surface to get our measurement. We just called that saturation difference for the purpose of this project. Now, as Patricia mentioned, we measured biodiversity in two ways. One was relative abundance, and how we got that is we did a 100 count. We looked at the samples from the net toes Brigitte described and counted the first 100 things we saw under the microscope, and how many of those 100 things were in different calcifier groups made our relative abundance. Then, using those same samples, we used molecular techniques to get species composition. Which species were there, which species weren't.
So, what did we find based on relative abundance? This is a really fun graph representing all the different creatures we found in each toe. So on the x-axis, you can see each net toe, which is in sequential order. So on the left, it's the earliest, and on the right is our latest net toe. And then denoted with a red M is our meter nets. Everything else is a Newston toe. Then on the y-axis, you've got the proportion of each taxonomic group that made up the whole sample. So out of 100, how many were of each group? And then color-coded is our taxonomic groups. Most importantly is that kind of lightish blue that says copepod. As you can see, there's a lot of copepods across the board, which means that our toes were really crustacean heavy. So we also did linear models in order to figure out whether there was a correlation between saturation difference and relative abundance, both overall and within these taxonomic groups. And we found there's no relationship between relative abundance and saturation difference, either within taxonomic group or overall.
So what does this mean? So this data implies that there could possibly be a general mesopelagic calcifier resilience to carbonate undersaturation, more so than we previously thought from previous literature. And also, we think that it's possible that this is because, as Brigitte mentioned before, our samples were really crustacean heavy. And as I've mentioned before as well, crustaceans usually make their shells largely out of chitin and not calcium carbonate. So it's possible that because they rely less on calcium carbonate to create their shells, they are less affected by it than other calcified groups.
So we've talked about our relative abundance results, but what about species composition? What you're looking at here is a plot of how different the species compositions were between different net toes. So there's a lot of variables that go into the differences in species composition. They can't all be put on a graph. So they're synthesized into two axes to make it simpler. The farther away each shape is from each other, the more different the species composition is, and the closer, the more similar the species composition is. So as you can see, there's no clear color gradient, but there is a shift from lighter on the right to darker on the left. They're color-coded by aragonite saturation difference. So we did a linear model to test this, and we found that there was a relationship between species composition and saturation difference. So what could this mean? We found a relationship. This could imply that some species within broader groups are more resilient than others. Some species might react differently to OA. In the end, we have to do more work to find this out. Other research has shown that some copepod species are more resilient than others. Other papers have found kind of mixed effects on pteropods in the face of ocean acidification. So that's what leads into where can we go from here?
Yeah, so future work. We want to emphasize that these results are really a starting point. There needs to be a lot more research on how ocean acidification affects these different taxonomic groups. We found that change in species composition, but we don't really know how those species compositions are changing and what species are affected. So further diving into that, it would also be really great to get a long-term data set going for this and redo these sampling methods year after year to account for variability in season and weather and that sort of thing, as well as different saturation differences over time. Also, an increased database of mesopelagic organisms and would be really helpful in order to conduct research like that, so that we can expand on this and create a more full picture of the mesopelagic environment. It's understudied, so building that library is important for deciphering some of these questions. And developing high seas policy. The High Seas Treaty specifically is a treaty among nations which facilitates collaboration and transfer of information from studies like this between nations. So just collaboration is really important to build these full pictures of ocean acidification. So continuing to build upon and make sure that the High Seas Treaty is encompassing that, so that that collaboration can happen. And Q&A, also our acknowledgements. Thank you to everyone who helped us. Yeah.
Great job. Let's start with the question if we have one in the audience here, and then we can hop to online.
Do you think that this research could be replicated on a different cruise track, and would it tell you a little bit more about the information that you found here? Or do you think it's important that it is replicated on in the same place?
Yeah, so I think one thing that's really interesting is on our cruise track, we only had so many variations in saturation difference. So the difference between the surface and the deep. I think other cruise tracks could provide really interesting information on whether that variation is larger elsewhere and whether that kind of larger variation is affecting calcifiers, maybe more significantly than it did here. Because again, we found some difference in species composition, but that's non-directional. We don't know if it was more biodiverse or less biodiverse. So I think kind of honing in on a finer scale on other cruise tracks could be interesting. It's just important to keep in mind that there will be different species.
A quickly related comment from Heather. She says, that was fantastic, clear description of OA. I'm curious about the range of saturation states these critters experience. What are the range, and are waters undersaturated?
Yeah, so carbon, okay, so aragonite saturation is typically described as anything below 1 will be corrosive to a shell. Like this shell will start coming apart. Our critters like to have an aragonite saturation above 3. We found a fairly significant, like the difference between shallow and deep was around like anywhere from like 0.9 typically to 1.5. So that's a pretty big difference for these guys. And they were all almost pretty much all under 3. We had one over 3. So it would be interesting to kind of look at cruise tracks with more like with higher saturation rates where they're happy to begin with. But yeah, so we found a pretty significant difference between shallow and deep aragonite saturation.
Yeah, I think also interestingly, when they dial vertical migrate, they encounter different saturation states as well. So some of the studies we were looking at actually found that, you know, if the saturation state is undersaturated at the surface and they spend more time at the surface for whatever reason, it can be corrosive even if they're not encountering it in the deep.
We have time for one more question. Sure.
I also want to quickly note, I don't know if you guys mentioned, but Brigitte drew all these cute little. Okay. Lula Diaz Villalpa. So sorry that I'm probably mispronouncing names. I went into this saying names, and now I have to continue. Asks, how did you all choose this topic? Why did you want to study this?
Okay. Well, so we were at the beginning of the semester given kind of like categories to choose from of like what zones we were interested in. I think we all came up with mesopelagic together, and that's how we got paired with each other. And then Patricia, I don't know if you want to talk more about like how we came up with this specific project.
Yeah. So we had the mesopelagic, you know, metabarcoding stuff as kind of a prompt, but then we were just kind of talking through it, and I think Resh has done some work with calcifiers before, so brought up that maybe that would be fun. And you know, ocean acidification is such a big point of interest with these creatures that we just kind of were snowballing and building on and came up with this as a final question. But yeah.
there were like calcifiers, and I was like, I know snails. And also shout out to Patricia, who does pre-med stuff outside of this, and now knows so much about calcifiers.
Great, let's thank the calcifiers.
A quick note for those on Zoom, if you are seeing your well wishes get deleted by me in the chat, I am taking pictures of them to show people later. Congratulations. Delete, there's just too many. Aw.
All right, so our next group before we have a quick break is going to talk to you about copepods and how they're responding to what they eat in the South Pacific Ocean.
Hello, everyone. Thank you so much, Sarah. My name is Ellis Konechny.
I'm Henry Penfold.
And I'm Charlotte Subak.
And we will be presenting about our research on copepod dial vertical migration and phytoplankton biomass in the South Pacific Ocean. So before jumping into our research and our findings, what is a copepod? You've heard about them in other presentations, but just an overview, copepods are small zooplankton that make up the foundation of the marine food web. Here on the screen, you'll see 4 examples of common copepod genuses that were found aboard the Robert C. Stephens as we were sampling. We have the Pseudocalanus, the Candicia, the Synchopes, and the Saphrodid. So not only do copepods make up a very foundational part of the marine food web, they also participate in dial vertical migration. Dial vertical migration is a very important process for copepods. During the daytime, they are living at about 200 meters in the mesopelagic and epipelagic water levels. And then during the day, they will swim up to the surface waters to feed on their primary food source, phytoplankton. This is very important as it plays into the biological carbon pump, which is essentially sequestering carbon into the deep ocean, very prevalent to the climate crisis that we are facing today. Thinking about the biological carbon pump, we are also looking at chlorophyll A. On the screen, you'll see a picture of chlorophyll A levels globally in the ocean. We are specifically looking at chlorophyll A as a proxy to look at phytoplankton biomass. Not only is this important to our research, but it is important to just generally look at chlorophyll A levels globally as the climate continues to change, oceans continue to acidify and continue to warm. It's definitely important to keep an eye on these things to see how it will affect copepod dial vertical migration and other processes in the ocean as well. So diving into our research, we have 2 hypotheses that we worked off of. Our first one is as surface phytoplankton biomass is increased, we would assume that copepod diversity would also increase as well. And then secondly, as phytoplankton biomass at depth increases, copepod dial vertical migration would also increase.
Yeah, so to test these hypotheses, the first thing we had to do is collect a lot of copepods. And we did that using the Houston net shown in the left photo here. And we deployed that net 47 times along our cruise track from New Zealand to Moorea, where we are now. And the product of a typical Houston tow looked something like that photo on the right. So that bucket just has a big pile of zooplankton in it. And from that pile, when we wanted to look at the relative abundance of copepods, we would do the 100 count. And identify 100 different zooplankton, and then we could look at the relative abundance of copepods to other zooplankton. And then because we were also interested in looking at copepod species richness and diversity, we would use morphological characteristics to identify all of the different copepods we found. And then we also did DNA metabarcoding, which allowed us to look at that in a different way. And then because we were interested in looking at the impact of phytoplankton biomass on deal vertical migration, we had to measure chlorophyll A, which we were using as our proxy for phytoplankton biomass. And we had two different methods of doing that. The first is on the left here, we have our hydrocast carousel, which we would send down once a day. And on that carousel, there is a sensor that could measure chlorophyll A concentration throughout the water column. And then our second method was using a flow-through system that continuously pumped water straight up into our lab. That's our lab there on the right. And with that, we could continuously measure surface chlorophyll A fluorescence. So getting into some of our first results, we have our copepod taxonomic diversity as measured by our DNA metabarcoding. This plot here, the y-axis is our copepod taxonomic diversity, and each dot represents a single Houston toe, and the closer dots are on the y-axis, the more genetically similar those toes are. And then the x-axis is our phytoplankton biomass proxy, which are our surface chlorophyll A measurements. So we expected that as the phytoplankton biomass increased, we would see that there would be a greater diversity of copepod species because there'd be more copepods migrating up to feed on this phytoplankton biomass. But this is a really surprising result. Our statistical tests found no relationship here. And there's a couple of potential explanations for this. It could be that the fact that copepods are grazing on phytoplankton at the surface is obscuring the relationship here. It might be that the higher phytoplankton biomass is in fact attracting more copepods, but then the fact that they consume all of this phytoplankton makes it so that our instruments can't really pick up that relationship. Some other potential explanations are there were quite a few confounding variables along our cruise track. In particular, temperature and salinity varied a lot along our cruise track, and those variables probably shaped the copepod community composition independently of chlorophyll A concentrations. So those variables might be masking the relationship.
So after looking at the overall relationship between phytoplankton biomass and copepod communities, we moved on to looking at the relationship between phytoplankton biomass and deal vertical migration of copepods. So what this graph here is showing is our evaluation of the rates of deal vertical migration. So basically what we expected to find was that we would have higher richness in our nighttime toes than in our daytime toes. And we evaluated that by pairing up the toes from the same day and calculating the difference in richness and using that as a proxy for deal vertical migration. But that's not what we actually found. So if you look here on this graph, you can see that some of our toes had actually higher richness during the daytime, or some of our toe pairs had higher richness during the daytime, or equal richness in the daytime and the nighttime, which was quite surprising. However, a possible explanation for this is that as copepod deal vertical migration increases, so does the migration of other deal vertical migrating organisms that can prey on copepods. So as the copepods migrate, so do the other organisms. They eat the copepods. Less copepods show up in our data, so that could be masking the difference in richness between our AM and PM toes. So then, as I was saying before, we were then looking at on our x-axis, phytoplankton biomass proxy. So we're looking at chlorophyll A here, but chlorophyll A is just a proxy for phytoplankton biomass since that's what the copepods are actually feeding on. And then our y-axis is looking at the taxonomic richness of copepods between the morning and the night. So that's basically a proxy for deal vertical migration. So what we're expecting to see here is that as phytoplankton biomass increases, we'll see an increase in the rate of deal vertical migration. However, that's not what we ended up finding. So this is what our results actually looked like. So we ended up not finding a relationship between phytoplankton biomass and rates of deal vertical migration. And this was a surprising result for us initially, but it makes a lot more sense considering the last two results that we just talked about. So considering the fact that we didn't see a relationship between phytoplankton biomass and overall copepod communities, and then also considering the fact that deal vertical migration wasn't showing up in our results, we know that deal vertical migration is occurring, but because of these confounding variables, it wasn't showing up in our data. So it also makes sense that we wouldn't have a relationship showing up between phytoplankton biomass and rates of deal vertical migration.
So finally, we have our relative abundance results. On this plot here, the y-axis is the difference in relative abundance between paired daytime and nighttime tows. And the x-axis is our phytoplankton biomass proxy at depth. So we expected for this relationship to see that as the phytoplankton biomass increased, the copepod relative abundance would increase because there'd be more copepods coming up to feed on this phytoplankton biomass. But this was yet another unexpected result. Yet again, we found no relationship.
So what are the implications of this? Why do we care? What do all these results and analyses, like, what does it really mean about copepods? So our results were quite consistent across our different analyses. We looked at metabarcoding data, we looked at morphological analysis, and then we looked at that in relationship to different measures of diversity. So looking at richness, relative abundance, and overall diversity of copepod communities. And across all those analyses, we consistently found that there was no relationship between phytoplankton biomass and copepod communities and copepod diel vertical migration. So we know that chlorophyll A and phytoplankton biomass is a really strong driver of diel vertical migration because there's a really strong body of literature supporting this. So our results aren't saying that this isn't true, but our results are saying that there's more going on here than we may have previously thought. And these are really important relationships to look into because the way that copepods respond to fluctuations in phytoplankton biomass has serious implications for the biological carbon pump, which Ella was talking about earlier. The biological carbon pump is how copepods sequester carbon, which is really important when we're considering resilience to climate change. So if copepods are more resilient to fluctuations in phytoplankton biomass, it means that we have a more stable carbon pump in the face of climate change. But what we're saying here is that phytoplankton is a factor in a really complex system. There's a lot of other confounding variables happening here. We have other physical variables like temperature, like salinity, like predation. Copepods are part of a complex ecosystem and a complex food web. And we need to evaluate all of these variables in relationship to one another to really understand what the resilience of the biological carbon pump looks like in the future.
So looking at future directions for our research, we really just need more robust science to continue to occur. We looked at temperature and salinity as different factors that might be affecting copepods and their ability to diel vertical migrate, but there are so many other processes in our world's oceans that are taking place as well. So really advocating for more science to occur at a wider ecosystem basis is very important to continue to understand not only simply just copepod diel vertical migration as they're going up and down the water column, but other issues that are now facing our oceans, specifically as we are facing threats from climate change. Thank you, and do you have any questions?
Great job. Let's do the same kind of pattern. Take a question from our audience here, and then we can move to online questions.
How much copepod biomass is there in the ocean?
You asked. So for everyone on Zoom, the question was, how much copepod biomass?
Yeah, it turns out if you took all of the copepods out of the ocean and weighed them, their weight would be equivalent to 16 billion double-decker buses. And you could fill 80 million Olympic swimming pools with them.
How fast are they?
Well, you ask, proportionally, they're 100 times faster than Usain Bolt. Size.
All right, let's go with one. We have time. We have time. We have a break, so we can go on the break a little bit. For some online questions.
Okay. Tom Subak asks, how do the copepods know when it's daytime? Does light reach that deep? And hello, Charlotte. See you soon.
I imagine it's more what's happening is they realize when it's nighttime, or they realize when it's daytime at the surface because suddenly there's light, so they go down. And then they kind of have a biorhythm where after that amount of time, they know to return to the surface.
Yeah, it's a natural rhythm for copepods, and the main drivers of deal vertical migration are predator avoidance and feeding. So they get hungry and they go up to the surface because they know the chlorophyll's up at the surface, and then they go back down to the depths because they don't want to get eaten because there's predators up at the surface.
However, it's also important to note that copepods live within the epipelagic and mesopelagic. In the mesopelagic, there's not much light that gets down there. So once again, that's reinforcing the idea that copepods really have this rhythm that they know when to go up and when to go down. It's also important to note that not all copepods deal vertical migrate. And some do sometimes when they feel like it, and some don't. Some do it pretty regularly, depending on their state, and there's more science that goes into that, but just something else that's important to know as well.
Yeah, so how does the sometimes migrating copepods, how do you think that might have affected your data set?
Yeah, I would say those copepods are generally the exception to the rule. Most copepods do deal vertical migration. So yeah, that might have had a small impact on our data because we wouldn't be seeing those ones. But yeah.
And some copepods also selectively migrate, so they migrate some days, but not others. If they've like eaten enough and their lipid sacs are full, they'll just stay down at a deeper depth. And then they only come up when they need to feed. So this also could have had an impact on our data, particularly where we're finding the inconsistent directionality with our daytime and nighttime toes, always expecting to find greater diversity at night, but not always finding that. That could be because not all copepods are migrating all the time.
Do different species like vertical migration, or were you able to test that, or is it something that
we certainly could test that. And when we were initially coming up with our project idea, we were thinking about focusing specifically on a couple of species. But for this project, we decided that we would be able to have a more robust data set and be able to draw more interesting conclusions if we were just looking at overall diversity across species, particularly because there can be difficulties with identifying specific copepod species. So we decided to look at overall richness.
Yeah, and with our morphological IDs and also our DNA metabarcoding, the lowest down we would go taxonomically was the genus level. So we could never really confidently say anything about particular species.
The sea state that you're offering affect
It definitely, the question was, did our sea state impact data collection? And it definitely did, because in order to evaluate deal vertical migration, we needed a daytime and a nighttime tow from the same day so that we could evaluate the deal vertical migration that was occurring within that 24-hour period. But often what would happen was we may maybe get a daytime tow and then we'd hit a storm and then we wouldn't get the nighttime tow from that same day. So it created some challenges when we were trying to pair up our toes and making sure that we were accurately pairing toes from the same 24 hours. That wasn't always possible. So it means we ended up with a smaller data set.
Yeah, out of the 47 Houston toes, we were only able to have 15 pairs, so we used 30 of those.
All right, well, let's take our 10-minute break and give our students a minute to shake out or prepare for the next set of talks, and we'll come back together at 10:30 and continue with our last few talks. All right, thanks for sticking around and coming back for our last two talks. Last but not least, we're going to start the next session with a group who is going to talk to you about siphonophore biodiversity. So I'll let them take it away.
Thank you, Sarah. Thank you all for being here today. I'm Georgia Akins.
I'm Brittany Durward.
And I'm Olivia Hines.
And today we're talking about barcoding the groovy goop, analyzing siphonophore biodiversity within the South Pacific using DNA metabarcoding. A gelatinous colonial organism that lives within our oceans are siphonophores. They're made up of zooids, and each zooid is
derived from the same embryo. They have different functions, but together they make up their elongated structure, which can be up to 47 meters long. They're due to their gelatinous structure. Taking samples and analyzing them is really difficult. Here's a cruise track that we took from Littleton, New Zealand to Moorea, French Polynesia, where we studied siphonophore diversity. We used molecular proxies of sea surface temperature, salinity, and subsurface velocity to measure their diversity within the pelagic oceans. Pelagic zone is the open ocean, and these communities are affected by climate change through influences of atmospheric distribution, wind direction, intensity of currents, and downwelling, which can affect distribution of these pelagic communities.
Moving into the biodiversity of siphonophores, because of their morphological structures and difficulty to identify, we were interested in using DNA metabarcoding to analyze their species composition on a molecular level. So we've heard a lot about DNA metabarcoding today, and essentially it is a widely used technique that allows us to look at genes on a very specific level. And this process involves extracting the DNA from certain samples and isolating specific genes, such as the cytochrome oxidase 1 gene, or CO1. And CO1 is really important because it is a part of mitochondrial DNA, meaning it can be found in essentially any living thing on the planet. So as a result, we have a very large database of CO1 genes that we can then take these patterns of our samples and compare them to other past species, such as siphonophores, and with a specific database developed by past studies that could allow us to go down to the species level and analyze species composition at that very, very small molecular level.
Siphonophores are a highly understudied organism, especially in the South Pacific. Gelatinous zooplankton are known for the evolved body plans and make good candidates for assessing climate change, especially within the South Pacific. We will be using these organisms to help tell us about climate change, and making baseline levels will help for future impacts that can be assessed as well.
Yeah, so for the objectives of our study, exactly how are we going to fill these knowledge gaps that Brittany just introduced to us? So under the basic umbrella of molecular analyses that Georgia introduced within the South Pacific Gyre, we hope to analyze siphonophore biodiversity and relate that to investigating physical oceanographic factors in this region, including sea surface temperature, salinity, and subsurface current velocity that Brittany also introduced. For the hypotheses of our study regarding the basic patterns of the physical oceanographic features that I just introduced more thoroughly, as we progress from Littleton, Aotearoa, New Zealand to Moorea, French Polynesia, we expect increased sea surface temperature, increased salinity, and decreased subsurface current velocity as we move in that northeastward trajectory. And then with the actual genetic data that we are collecting, we are expecting increased siphonophore biodiversity at the beginning of our cruise track near coastal Aotearoa, New Zealand, largely due to the impact of the EAC, which comes down just along the east side of coastal Aotearoa, New Zealand, as well as that interacting with another western boundary current. So both of those bring really warm, nutrient-rich water into this region, which is great for siphonophore growth and biodiversity. And then as we move again northeastward toward Moorea, French Polynesia, we expect decreased siphonophore biodiversity because of that increased salinity and decreased subsurface current velocity. Those compounding factors lead to decreased siphonophore mobility, so the distribution and biodiversity will likely be lower. And then for the purpose of our cruise track, we divided our bioregions into these three regions where we were able to collect siphonophore DNA: coastal Aotearoa, New Zealand, Southern South Pacific Gyre, and Central South Pacific Gyre that you have seen in previous presentations today.
We used two types of sampling methods. The first one was the Newston tow, which was deployed for 30 minutes, and it took surfaces on the surface. It took samples on the surface. The second was the meter net, and it was deployed to 300 meters, and it took samples for us that were below the surface.
To specifically analyze the biodiversity, as I mentioned earlier, we were interested in using the molecular techniques. Specifically, we use DNA extraction to take the DNA from these species, but specifically from their pooled samples. So pooled samples, or the goop, as we like to call it, is, as you can see here on the side of the slide,
It is essentially everything that comes into the net as we do these toes. So there are more than just siphonophores in these toes, but they can be filtered out through later computational sequencing. And that involves extracting the DNA and isolating that CO1 gene. We can then take that CO1 gene and run it through PCR or polymerase chain reactions to amplify it. And essentially, this just creates a bunch of replicates of this gene, making it easier for us to read and eventually making it easier for us to decipher the presence of certain species.
So moving on to our results and discussion, the first thing that everyone wants to know, taxonomic categorization, what and how many siphonophores did we find? We found a total of 33 species, but I have 32 written here because as the Physalia group presented, Physalia are siphonophores, but because they had the focus of Physalia in that group, we decided to exclude this data from our data set. But these 32 total species excluding Physalia spanned 3 suborders of siphonophores. And just for fun, we found 2 really rare species of the suborder Cystonnectae. And one of these species was actually discovered in the past 5 years in the mesopelagic off the coast of South Africa. So crazy species distribution here. This would be really interesting to get more of a worldwide distribution of specifically this suborder.
All right, moving into the data. This plot may look a little bit scary at first, but I can assure you it is not. This is what we like to call a non-metric multi-dimensional scaling plot. And this essentially allows us to look at genetic diversity amongst our species, amongst all our samples, but only on 2 axes. Like we've heard today, genetic diversity can be really hard to analyze because there's so many different axes, but putting it through this statistical test allows us to look at it visually and a lot easier. And something that is really important to note is that species and plot points on this plot are going to be more genetically similar when they're clustered together, and the ones that are further apart are going to be more genetically different. And so as we can see with our data, it at first looks a bit scattered. It doesn't really look like there's a lot of clustering. But when we look at it closer, we actually see that there's a clear delineation between general locale amongst our samples, specifically within the southern South Pacific Gyre in blue and the central South Pacific Gyre in green. The samples from the coastal Aotearoa New Zealand area are more scattered within the southern South Pacific Gyre, so they are not as clustered within their own category. It is important to note, though, that it is important to note that when we see these clusters, we cannot specifically say they're more biodiverse or less biodiverse. It is simply a matter of species composition. And moving on to our abiotic factors that we're interested in, temperature, salinity, and subsurface velocity, when we put these on these plots, we see some very similar patterns. With temperature, we once again see a clear delineation between the southern South Pacific Gyre and the central South Pacific Gyre. And with subsurface velocity, we also see this separation, with it being a little closer and the subsurface velocity not varying as much. But when we get to salinity, we once again see the same pattern, but this difference is a lot bigger. And it may not look like it on this plot, but this difference within species composition is really significant, even though it physically might look like they're a little bit close. It doesn't look like they're that far. But when we run it through statistical tests, we actually see that there's a clear impact of salinity on the species composition differences. And this is important to note because salinity does change drastically throughout our cruise track, meaning that as we move northward, the water becomes more and more salty as a result of increased evaporation. It is important to note, though, that NMDS plots cannot be scaled for directionality of species composition and biodiversity. What can be scaled for directionality is species richness. And so, as we can see on this plot, we have plotted species richness from our cruise track all from Aotearoa New Zealand all the way up to French Polynesia. And essentially, species richness is derived from the number of species we found within a tow compared to all the species that we could have found. And as we can see, there is a shift in coloration as we go northward. So we have a lower species richness in the southern part of our track, and it increases as we move closer to French Polynesia. And when we overlay this with our general locales, this difference is extremely clear. And although this does go against our initial hypothesis of seeing increased species richness near the coastal Aotearoa New Zealand area, it does still show a dramatic impact of general locale on species richness, even though it was in opposite places than we expected.
So future directions, what implications are there for this lovely genetic data that Georgia just presented for us? So going back to the beginning that Brittany mentioned, most of our
implications are framed within the perspective of climate change, unfortunately. Of the 3 abiotic factors that we looked at, the most applicable within the South Pacific Gyre specifically is actually subsurface current velocity. Once again, due to our friend the EAC, which is totally not tubular, the EAC is expected to increase in velocity by up to 20% in the coming years, which is good for siphonophores because that's more warm, nutrient-rich water for them, but bad because we currently don't have those baselines of zooplankton community distribution in this region. So we have no baseline science to compare communities as this current speeds up. Also, you may have seen news articles similar to this. Jellyfish are taking over the world. As the Physalia group mentioned, jellyfish and siphonophores are very closely related. So it's expected that siphonophore populations will increase along the same magnitude that jellyfish populations will. Again, we currently have no baseline studies for this in the South Pacific to be able to compare this headline and see if it's true in 5-10 years. And lastly, for future studies in the South Pacific, specifically with zooplankton communities, we propose that environmental DNA or eDNA techniques are evolved to incorporate the CO1 metabarcoding because it is universally applicable and because this would be a relatively non-invasive, easy way to expand our molecular database in this region.
We would like to thank all of our partners who helped us with the research.
Any questions?
Great, let's open the questions to the room and then we can go on.
Most nervous part for you right now is your dad asked me a question.
Yeah.
But first, I'm so proud of you, Jordan. This is
Thank
Everybody. One of the graphs you showed the cruise track, there was a gap in it. Taking a break or yeah, right there.
Let's go back there.
Yeah.
So
breaks. Well, if you look closer, there's actually 2 breaks. There's one closer to coastal Aotearoa New Zealand and one southern, so the central South Pacific Gyre. This was a limitation of the weather we encountered, which, as we all are familiar with, the really intense wind and seas that prevented us from conducting science for quite a while. Yeah, the 60-foot swells, 100-knot winds, laser sharks, you know, all those things. But yeah, we weren't able to conduct tows during that time.
Being hoped to for 3 days isn't exactly conducive to moving at a speed of about 2 knots to conduct news, unless you were moving 2 knots sideways, which luckily we weren't.
Yes, we have to slow the boat down to 2 knots, which is about 2 miles an hour.
Any questions?
Do you have any insights or ideas on what could happen to the ocean and the ecosystems within it if siphonophores do take over?
Yeah, I think it would be a problem of there would be too much prey for certain species to keep up with. Siphonophores are primarily eaten by nudibranchs and like xanthinid snails or bubble wrap snails. So essentially there would just be too much food and it would probably cause a spike in some of those predators, and then eventually they would fall off because there would be not anything to eat those secondary predators. So it would just cause a whole bunch of disruption.
But the birds would be very happy.
Birds would be there'd be a lot of birds.
And abiotic factor wise, that would probably create more oxygen minimum zones near the surface, just because of increased respiration of all of these organisms, increased predation. So, and it could be good for carbon sequestration in a way because you have all of this gelatinous carbon that is now sinking, but that will definitely have a top-down effect as well.
What do you expect to find
What was the last bit?
What would you expect to find if they made a cruise track all the way to that?
Oh, okay. Just for Zoom, the question was, what would we expect to find with siphonophore biodiversity if we continued all the way up to the equator? I would believe there would likely be an increase in siphonophore biodiversity actually because of equatorial upwelling, bringing cold, nutrient-rich water up around the equator zone, as well as the increased temperatures there as well.
No, that's something. Online question from Michael Muse. Was pH not a variable in your project due to consistent pH throughout the regions? Yeah, basically, why wouldn't you maybe consider pH?
The reason why we didn't consider pH is because we were already interested in looking at quite a few other variables, temperature, salinity, and subsurface velocity. Although pH, I think pH would be a very interesting factor to look at, since siphonophores are not calcifiers, as we heard from some of the previous projects. They do not, they wouldn't have that same impact with like carbonate saturation, but it would still be interesting to look at how pH could affect their gelatinous structure.
pH was originally on our list. We had a big brainstorming session in Madden on the whiteboard in the living area, and pH was on the list. And I believe we actually swapped it out for subsurface current velocity when we realized there would be the ADCP on board, just because siphonophores are planktonic organisms for the most part, so the current would have significant effect on their distribution.
Great. Well, we have to move on, but if there are more questions, we can ask them at the end.
So good. How many salts make up a salmon? Oh, I got all of them. Oh, it's really handy.
I just framed your just.
All right. Oh, my voice just. So we are going to close with a group that is going to talk about myctophids, our fun little fish, and how they respond to low oxygen across the South Pacific. So thank you.
Thanks, Sarah. I'm Andrew.
I'm Jamie.
Zach.
And I'm Elle.
And we're going to be presenting on diversity, under diversity, myctophids, and low oxygen environments. So what is a myctophid? Myctophids are small deep sea fish that are highly abundant in all of the world's oceans, where they make up a very important part of oceanic food webs. They're also known as lanternfish, and they get that name from these photophores, which are specialized light-producing or bioluminescent organs that emit a blue light. Those photophores, the arrangement of them can also be used to identify them to the individual species level. There are an estimated 250 species of myctophids worldwide, so they're a very diverse family, and they form the majority of small deep sea fish, which combined have an estimated biomass of 10 billion tons, which I know is a very abstract number, so I've translated that into double-decker buses. I know it's much easier to understand. Myctophids are diovertical migrators. We've heard about that before today. So they spend their daytime down in the mesopelagic, which is a layer of the ocean that spans about 200 to 1,000 meters deep. And at night, they come up to the surface to feed and avoid predators. That diovertical migration means that they are an important carbon transporter as they move nutrients from the surface down to the depths. And it also means that they have to pass through these oxygen minimum zones, which are areas of low oxygen concentration. Also, because they spend so much time at depth, despite that super high abundance and diversity, they're fairly poorly studied.
So studies have shown that as climate change worsens, OMZs are expected to expand and intensify. So our hypothesis is that as OMZs intensify, biodiversity is reduced. Myctophids are fairly resilient to low oxygen environments, and they're equipped with certain physiological mechanisms that allow them to stay in these low oxygen areas. There isn't extensive research in the South Pacific, specifically on myctophid biodiversity. So our hypothesis is based on last year's cruise track, which was similar to ours, S314.
And so our study compiles data from both last year's cruise track as well as this year's cruise track. For sampling of abiotic factors, we deployed dissolved oxygen sensors and depth sensors down to depths of up to 600 meters. And this allowed us to obtain a dissolved oxygen profile from which we could see where the OMZ was located, as well as what the minimum dissolved oxygen concentration was within the OMZ. And for sampling the myctophids themselves, we deployed newsstand nets and meter nets, which you've heard about from the other groups. We deploy them at nighttime when the myctophids are close to the surface, and so we're actually able to catch them. And once caught, we first identified the myctophids to the species level using phylophore identification under a microscope. And then from this species abundance data, we calculated the Shannon Biodiversity Index for each of the sampling locations in order to quantify the biodiversity at those locations for further analysis. And then after identification, we extracted DNA and sequenced the whole genome of select specimens for genomic analysis. And so here you can see a map of all of the specimens that we collected over both cruise tracks. The circles, the size of the circle represents the number of individuals caught, and the color of the circle represents the species that was caught. And over both cruise tracks, we caught 221 individuals, which represented 22 species across 13 genera. Over half of these were all the same species, which was Gynectes barnacii. And on both cruise tracks, we sampled across a range of dissolved oxygen and OMZ depth. And so here on the left, this is what we sampled for dissolved oxygen. You can see that as we move towards the equator, more north, we saw a lower concentration of dissolved oxygen. And then for OMZ depth, as we move towards the equator to the north, we saw deeper OMZs generally. And so taking these OMZ variables of dissolved oxygen and depth, we compared them to the biodiversity that we found at the stations, and we saw as the dissolved oxygen concentration decreased and OMZ depth increased, that there was generally more biodiversity in these myctophid communities. And this is pretty interesting since it contradicted our original hypothesis based on last year's analysis. But it does more closely align with what other research on myctophids in different regions has shown, and that they are more resilient to oxygen minimum zones. And it's important to note that these physiological adaptations that they have are likely shorter term adaptations, and we would need to do more longer term research in order to determine if they will be impacted on the scale that like climate change intensifying the OMZs, like whether or not that would impact them overall. And it's interesting also that we saw that biodiversity increased rather than just staying the same in lower oxygen environments. And a proposed mechanism is that the competitors and predators of the myctophids might not be as resilient to low oxygen environments. And so therefore, in the low oxygen environments, there's different selective pressures that could allow for more biodiversity in those areas. But also more research would have to be done in order to really tease apart those ecological relationships and determine what the basis for this relationship that we saw was.
Thank you, Jamie. Let's get into the genomics. Out of the multiple different taxonomic groups that we were able to find, there were two unique species that we were able to subset and look at genetic data for, G. barnacii and D. atlanticus. For G. barnacii in particular, we were looking and interested in the principal component analysis. Similar to what you guys saw, I'll give a quick overview. We split into two. A principal component analysis allows us to take high dimensional data and boil it down to something we can visualize relationships with. We have two principal components, the percentages of which telling us the total variation within the population. The thing to note here is you see dots that are close together, they're more genetically similar. As you can see here, we have two cruise tracks. Our circles in 2025, the squares being 2024, along a specific latitude. And specifically, we have one cluster that appears to show up. Not only is it between the 2 years that you can see it, but there's a latitude correlation just based off of the color there. Our lineage analysis also showed this. From south to north, these are the lineages that pop up, three specific lineages that were found along our track. And as we move south to north, we saw these populations or these lineages within the specific species change, going from the blue and the green into our pink. One thing to note here too is that each bar that you see here denotes one specific lineage, one little tiny bar. Most of the times when you look within a species, you'll see lineages that cross and mix. As you would expect, it's within one species. But within G. barnacei, within our entire cruise track, these lineages were distinct and set. Unlike G. barnacei, where we were able to get several species to sample for our sequencing, D. atlanticus, we were not. And that may be in one part to, as you guys heard, the crazy cruise track we had, and maybe some less than stellar sailing on our part sometimes, with the crazy winds. But when you look at the data between both years, there's one specific spot where D. atlanticus shows up, and that seemed to have a distinct correlation. Just one thing to remember as well, within the specific region, this is the South Pacific Gyre, and so you have the currents that go outside the gyre and that run along the west-east section right here. For some reason, D. atlanticus like to set up shop in this one section that's specifically within the current. Something that was unique. Not only within the specific gyre itself, but within this population, we saw unique latitudinal gradient, similar to G. barnacei, where as you move south to north, the lineages within this species changed. Similarly, one specific tick here represents one sample with its unique lineage, with our outlier being sample 218 that had almost the exact 50-50 split. Again, those populations can be seen here as we zoom in, population 1 and population 2. So why is this important? Between the two species that we found unique and incredible population differences within each. With G. barnacei, we were able to find them across our entire track, but found lineages that were distinctly based on latitude as we moved along our track. With D. atlanticus itself, it was completely opposite. We found them only with one specific spot between both years. But again, there was a unique latitudinal gradient and relationship that happened within that species. These are things that we would not have learned about if we had not taken this data, if anyone had taken this data. We are lucky enough to be part of this program that partakes in incredible cruise tracks across the world that take us into unique locations that, for instance, like the South Pacific, that have barely been studied. We had, as you saw in different presentations, there's barely any baseline data in this area. And with more data in the future, we'll continue to learn more about these incredible and unique relationships that are unexpected. Like, for instance, the Atlanticus setting up shop in the middle of a giant current across.
Okay, so concluding, we found that myctophids are resilient to low oxygen environments. Genetic analyses revealed population structure within our most abundant group of myctophids, G. barnacei, and population structure could suggest that they're more vulnerable as OMZs expand and intensify. But again, as Jamie mentioned, the long-term implications of OMZs are still unknown and further research is required. Additionally, there are more intense and expansive OMZs in the Central and North Pacific, and how those areas, how myctophid biodiversity is affected by those areas is not something that we study. And then in the future, long-term, as I said, long-term implications of OMZs are uncertain. And in terms of policy implications, myctophids are carbon transporters that, as Andrew mentioned in the beginning of the presentation, and because of their large biomass, they have the potential to be fished on a larger scale. So as myctophids are studied over time, it can influence marine policy, marine science, and public policy.
Thank you, everybody.
Great, we'll do our short question and answer for our myctophid group, and then we can open up to broader questions from all the groups. Any questions in the room?
Go ahead, Rush.
Yeah, so you guys mentioned in your conclusion that population structure might make myctophids more or I guess more at risk. Could you explain a little bit more about that and like what kind of future work you'd want to evaluate that further?
I'll take it. Actually, go ahead.
So one thing with that population structure is we don't see a lot of gene flow between the different populations. So you have the issue of these populations then being fairly isolated. So you don't get those passage of different genes that might make them more resilient to change between the different populations. So if one population is more resilient than the others, those genes don't get passed to the other populations, and those populations are more vulnerable to those long-term changes.
Also, one thing that was unique specifically in the Atlanticus, you saw that was one specific spot, and a spot that we wouldn't have expected. And for if you're looking at a map or any other reason, you wouldn't expect there to be one specific species in that area. Again, more data may show later in time that there's, different subsets, but that species, there's obviously a reason that that's there, and until we know that reason, we can't do a good job to protect it. So that's going to be something that in the future, as more data comes along and we understand more, it's going to be really important, especially as climate change continues to affect the region.
Okay, Eric Lorraine asks, is there a correlation between the water temperature and oxygen level and effects on the species population and diversity?
Yeah, I mean, we didn't specifically do a statistical analysis with the water temperature, but because we did find that as you move closer to the equator, there's changing effects on the OMC levels, that there's also a correlation with the temperature, because as you move closer to the equator, it gets warmer. But that would be something that would be interesting for future study to see.
Also, I can add on a little bit to that, myctophid adaptations can be directly because of changes in temperature, but they can also apply to low oxygen environments.
Any more specific myctophid questions?
Adaptations that have that allow them to live in the
They release heat shock proteins, and that's the extent of what I know about them. That they allow that lets them remain in the low oxygen zone.
Part of that is that there are just not a ton of specific studies on those. That there's a handful on the actual physiological adaptations that allow them to do that. It's mostly just we know that they can hang out there, so they must be adapted to it.
Yeah.
But part of how exactly that works. So
You know anything about how predators handle OMCs?
So the question was, how do myctophid predators handle OMZs? There have been, this is again an issue of lack of studies. But generally speaking, most myctophid predators prey at them near the surface, which is part of why they diel-vertically migrate. So it's probable that they're less adapted to low oxygen environments. But again, the exact physiological adaptations.
They're also predated on by a lot of different things. So other fish, squid, and penguins even. So it's hard to say generally if all of them would be impacted. And there are a lot of studies that show that other fish are not quite as resilient to OMCs, and so we would expect there to be some kind of influence on other organisms that hang out with myctophids.
Again, further research more.
Yeah.
It tends to be a similar theme in this. Cool, wonderful, thank you.
I'm gonna keep it on the slide because it's lovely. One of our swim calls when there was not a gale.
Well, this was right before. Sister, I believe.
But I would like to open it up for all, a sort of general question and answer session now. So we can have Kayla monitor online or have questions from the room. And if somebody's asking your specific group where you want to come answer the question, I asked you to come up so that the microphone can actually hear you from this projection computer. So do we have any? Yeah, Rick, Captain Rick Miller.
Yeah, so all of the patients talked about sampling techniques, and we had surface net nodes, meter net nodes, all the vessels that were peanuts, and then also the boat cruise scale and stationary. I wonder if there's research and considerations for vertical meter net code on the boat too, say for deeper and also in the column. And also there's thoughts about doing temperature you can capture samples of specific depths without coming out to the entire layer of the ocean.
Yeah, so if the microphone didn't pick it up, that was our amazing captain who sailed with us this journey. And he was talking about potential different kinds of sampling methodologies. So we have the standard net tows that we do are sort of across a more horizontal space, whether that's at depth or the surface, and talking about doing net tows that might be in a vertical space, so down to a depth and back up, where you're integrating that signal of what's in that exact space, not maybe over a nautical mile, or doing nets that can open and close at specific depths. And those are definitely really interesting sampling methodologies, and would, I think, do an interesting add to our biodiversity studies by letting us think about what's exactly at depth. And I might bring Kayla Gardner up here to talk about. Sorry, I'm monitoring the chat. No. She's monitoring the chat, but to talk about like the strengths of something like a Tucker trawler, or Kayla has used mock nest, which is a different kind of setup. We usually use on much bigger ships, but that can open and close at certain depths, and what that might add to our biodiversity studies. Kayla's our postdoc.
Yeah, hi everyone. I'm a postdoc with SCA. I recently got my PhD in mesopelagic zooplankton and fish. So, yeah, so I use something called a mock nest, which is a multiple opening and closing net system. So ours are 10 meters squared. You know, they could, you know, span the length of this room. They're so big. And we have up to 9 nets on ours. And so from the ship, we can tell them when to open and close, basically, which allows us to say that a certain sample is from specifically 1,800 meters. And then as we go up, 600 to 400 and stuff like that. And that's really important, as a lot of these guys really perfectly communicated, because there's so much of this migration happening. And so we don't know actually which species necessarily migrate every night. As our copepod group got into, sometimes they don't feel like it. And so it's really important for us to know where we're getting our samples when these species are creating or are participating in these huge vertical distance changes. And so it's really cool to get specific depth information, because then you could also compare it to specific depth information on our hydrocasts and get an even more nuanced view about how these physical factors are affecting these specific organisms at these specific depths, and also which organisms are migrating and to what depth. If we knew exactly what depth we caught our myctophids at, or how deep they were, we would be able to know if they're hanging out in the oxygen minimum zone, or if they have to transit through it, or if they're sitting above it sometimes if it goes deeper. And so, yeah, so it's really important. You can get so much other information from these kinds of depth specific tows. And they're really cool. They're just a lot more complicated, which is why there aren't mock nests on smaller vessels. They're probably the joy and also bane of most scientists who use them's existence. So yeah, but they're really cool tools, and it's really cool that a boat like this would also be able to get that, because I on my cruises didn't go to places that are as understudied as this. So I think this cruise was really cool, because I wish I had samples from this region when I was doing my PhD. So yeah.
So thanks for that great question, right? Are there questions online, Kayla, that we want to address?
Okay. Aaron Bryant asks, how would you like the UNBB&J science-based conservation communications to talk about Physalia and how to protect them? I think other groups could also weigh in. If they have.
All right, come on down.
Oh, okay. The question was, how would we want them to, like, inform people about it?
yeah, like how would you like them to communicate to others about your group? And like that's why I said maybe other groups can win if they want.
Yeah.
With their specific groups.
So I think a big part of this is the church study, because our data is being added to the church study, and they're really like the pioneers of Physalia speciation, even though other people have like thought of it before. But just I feel like a lot of people don't. Everybody knows that the ocean exists, obviously. Probably. But I feel like the extent of the ocean, like how big the ocean is, what is in the ocean, is really understudied and misunderstood by a lot of people. And I think that is the whole goal of the biodiversity beyond national jurisdiction, of like sharing this genetic information. I think the goal, I believe, is to create a database with all the genomic information that can then be used for more extensive science and more combined science.
So like as an example of that, a lot of pharmaceuticals and just things that end up in medicine come from the natural world, like they're not designed. So neurotoxins are one thing that has been used in medicine a lot, and as we've noted, Physalia sting. That's kind of the thing they're most well known for. So having, first off, 4 different species that probably have it's entirely possible that they have 4 different neurotoxins. That's, or maybe more, given that they might have different kinds within one species. That's more opportunities for possible medicine development. That's the kind of thing that like we're hoping that a larger ocean genome of all of the critters we've talked about, Physalia is just the example that I've done the most thinking about. Those are the kinds of things that are useful to humanity. So if we can make them accessible and give people the opportunity to use them, good things can come from that.
And one of the like main goals of the High Seas Treaty is to share this information with like everyone, even like with landlocked countries. And so like that's another thing that I think is really important, that like everyone has access to this information, and it's not like kept within like the countries that are like rich enough to do the research on it, that this information is actually shared with like the entire world.
Great. Any other groups want to add to that for your particular species study group? You got to come up.
Get up there, bro.
Talk to
for
the,
all DBM related. I want to just go up there.
that's like everyone.
Make it circle. God.
Our secret thing.
Yeah. Oh God. Okay. Well, I think one thing that was noted, I guess, and it's perfect because it's between all groups, is that every one of us talked about there's need for more science. There's nuance within the species that we have. There's complexity that was unexpected, and we had our hypotheses based on other science and other places and other areas, and we were often wrong and completely off, and new things came up. And whether you want to take it on a uses base for humanity or just for a protection base, as we just in general, we're in such an interesting phase in kind of the global environment where we're watching things actively change, let alone the ability for people to interact with and also affect these really varied and complex species is ever growing. If we don't take that into account, let alone begin to study these species and have a baseline for things that we may miss, things that may change, we'll be losing incredible data, incredible understanding that may allow us to protect them in the future. Any way you slice it, there's interesting factors here that can be saved and may potentially be lost over the next generation. And for all of us here, I think we want to see that not happen. So yeah, if we run another cruise track, let us know.
So one thing that we found in like the TOFA group and the Physalia group is like hidden biodiversity that wasn't known about before. And you can't protect biodiversity that you don't know about. So with myctophids especially, they're not commercially fished right now.
Yeah.
It's not profitable, but that's yet. And they have been looking into that, especially for things like aquaculture, fish feed, or pharmaceuticals or cosmetics. And so it's the sort of thing where if we don't learn more about these communities and these populations, we might not be able to protect them effectively and get the benefits that we could get from whole ocean genomes without destroying.
I wish I'd known you guys were going to be so insightful with your answers and had ended on that question.
I'm gonna make the students bigger.
Okay.
On Jared.
Kristen Chinesky asks, does light have any other important influences on these organisms, or does it just provide a place, a reason for them to need to kind of hide from visual predators? Like, what do you as the DVM groups think maybe light would have an impact?
Every group.
Does light have any other important influences on the DVM organisms, even secondarily?
Well, yeah, I guess with copepods, light kind of drives everything because they feed on phytoplankton, and without light, we wouldn't really have phytoplankton because they're doing photosynthesis to survive. So that's, I think, one big one.
You can also take that another way. As we talked about, our myctophids have photophores, and so are actually, as I'm looking at Kayla, are identified that way. And we actually had a unique and really interesting example of one species that we thought was one species and looked exactly the same as G. barnacei. And when we did our genomics, it was way the fuck over here, and found out that just by a little tiny altering of where the photophores were placed, it was a completely different species and genus. Yeah. But although that's not daylight when you think of it, those photophores are used to communicate and for different species to hunt, and especially we talked about being down in the mesopelagic for life. That's a whole other world. And of course, I'm assuming most of us have seen clips or pictures of David Attenborough and Nat Geo stuff where it's the incredible ways light's used down below. And God, how incredible would that be to eventually want to be able to study that and see that. But we can only know that from the species that we bring out and tend to be dead already. But we jiggle a little bowl, we'll see light.
One other thing with myctophys and light is that we weren't able to actually get into it with our study, but some other studies have found that especially what depth they come up to at night is really strongly affected by moon phase and how much light is coming from the moon. So light in general can just affect where you find these different organisms in the water column.
I think also just thinking about, we have our myctophid group and copepods and our calcifiers, like all of these organisms make up very foundational parts of food web. So if we think about this very holistically, light contributes to their lifestyles and their life cycles. And so it's an incredible, not only does it penetrate down to certain levels, but it also affects entire life cycles and then therefore the entire food web.
I mean, yeah, most of what myctophids eat is copepods. So whatever affects the copepods affects the myctophids.
Which then will affect bigger organisms and megafauna.
And copepods are calcified.
Yeah, that's a question.
Yeah.
Yes, we saw a lot of talk about keeping up and down. We have some really great full moon nights. Did you notice differences in the migration? We have big full moons over the moon.
Remember when we had a meter net, if there was a full moon, what I would say? I was going to bed early.
You guys want to repeat the question for the computer?
Yeah. Cap asked how full moons and moon phase affected our net cast, particularly on full moon nights. We have certain nights where no clouds out and you could see everything. Was wondering how that affected our internets or our net toes. I think that's data that we didn't specifically look at, but as Kayla mentioned, is definitely a larger effect. I don't know if anybody was able to look at it within their own data.
Just observationally, in our Newton toes, we would still catch myctophids, G. barnacei. One of the reasons we caught so many of them is they seem to be really tolerant to high moonlight, which was really crazy for me. I didn't think that would happen. But like I said, I would go to bed early if there was a meter net because the deeper myctophid species were way deeper on moonlight nights, interestingly.
So moonlight means Kayla gets more sleep.
This also has to do with light, but I know that these are a lot of pelagic species, but are there any studies or insights about if things, like if there's like a deep shelf near shore, if like light pollution would be an effect of species, or what's that look like?
It would probably affect like because some of these predators are visual if light is in the water. I know as far as from a megafauna perspective, with sharks and sea lions, it can mean that, you know, the sea lions naturally go into the water when at night to hunt because the sharks can't see them, but if there's light pollution, the sharks will hunt them at night. So on a, you know, smaller critter scale, I could imagine that the same thing could happen if the light pollution was substantial enough that the light would penetrate enough, at least at the surface, to be able to see a little bit better.
Even just anecdotally from us, you know, sometimes, especially when we were at anchor, we do a call squid TV.
Yeah.
Just shine a really bright light over the side of the ship. And I've seen like needlefish hunt using that light from us. So that is something that I would definitely expect certain species utilizing that light from human sources.
On Big Valley, that one thing too.
Yeah,
that was cool.
Question for the Physalia group: what is the mechanism for the sting, and what is the toxic?
Somebody made the mistake of asking a stinging cell question. 3 hours.
We got a whole presentation on it last night.
Okay, yeah, it's true. I did give a presentation on this last night. All right, I'll try and keep it quick. So, cnidarians, which is the whole phylum that Physalia and jellyfish and corals and sea anemones are part of, all have stinging cells of a couple different kinds. Physalia and jellyfish and everything that you know that hurts only has one of the 3 kinds called nematocysts. And so what it is it's this little capsule that sort of kind of looks like a grain of rice with a harpoon attached to a string in it, essentially. And so when you get stung, so when you touch one of these stinging cells, the harpoon flips inside out in order to shoot out. It's one of the fastest things that happens in nature. It's one of the fastest things in the animal kingdom. It's a nightmare to try and get on camera because in order to take enough frames to be able to see what's happening, you actually don't let enough light into the camera to be able to see it, which is an ongoing problem of mine. So if anybody has a solution for that, let me know. But you flip this harpoon inside out, and then a little tube with a whole bunch of neurotoxins in it gets through whatever skin the harpoon got through and put into the fish or finger or whatever it is that came in contact with that stinging cell. So Physalia have a pretty gnarly sting, as people are aware, so they've got a number of those fairly large stinging cells with a whole bunch of neurotoxin in them, with a really cool system to get it into you.
You didn't catch, she does work on this outside of
Yeah. This is what I do back at normal school.
Normal school.
Not school. Yeah, that's cool.
Okay, we have one more group specific question, and then maybe some general ones. Copepod group.
I don't think we should talk about this.
I didn't listen.
Like a little choir box.
Satya Advani wants to know, were there any color difference in your copepods, and do you have any information on if they migrate or behave differently in those color groups, or anything about color in copepod?
There's definitely a ton of color variation. I think some of our photos showed a bit of that, but a lot of them are kind of dark gray, not too interesting looking. But then there's also these brilliant blue ones, the Stafford copepods you would pull up a lot of the time. I think those are the main colors we would see. We didn't actually do any analysis of whether they're migrating differently based on those colors. But yeah, it'd be interesting to look into.
Yeah, and as we pulled up our buckets from our new student nets and our reader nets, we would often notice that a bucket would have a certain color overall. So we'd be like, oh, this is a really red bucket, or this is a really blue bucket. And we didn't do analysis of why that is, but it was definitely a pattern that we noticed as we were looking at buckets from our toes.
And it would also be interesting to look at for future research. Our brilliant blue ones were a lot bigger than our more transparent ones.
Right, for all the groups that had multiple years of data, there's a question from Ruth Muse. How does your data compare to last year's sampling? Did they collect more or less? Anything else you noticed between those?
You don't have an answer for this.
Okay.
Tell your mom.
Go back for
Last year they caught a lot more samples in the central South Pacific Gyre because they didn't have a gale there. And so that was different. One thing that was similar is that they also didn't catch any Physalia in the tropics. Which we also didn't, which is interesting because they do live in the tropics. So that's something that's cool. I don't really know why that happened.
Our best guess, there are fewer of them there, probably, than there are a little bit further south, but they are there. We should keep going because one day we will catch one.
With the myctophids group, so you saw in the D. Atlanticus that we had, the two different populations that we had were between years. Like one population is what we found and one population is what last year's groups found. So there is some differences there. There were also several species that last year they caught and we didn't. And I have a slide for this actually in our photo slides, unfortunately. But if you look at where sampling stations occurred, you can see that during gale number 2 for us, where we didn't collect any, at the same latitude anyway, last year's crews did do stations there, but they didn't find any myctophids. So there are just interesting things there. There is also one species that they found exclusively like along the Chatham Rise between coastal Aotearoa New Zealand and the Chatham Islands, and we didn't do any toes there. So we didn't find that species at all.
AKA more death, more death, more death.
That was another gale.
Yes, that was gale number 1.
Did you have a related question?
Yeah, so my question was, if anyone remembers from last year's data about wind, wind speed, especially when you got to the tropics, was it similar? Because I know the wind really drops out and so you get tropic after.
We were not provided with the wind data.
It would have. Well, okay. I didn't perform any analysis.
Comparing analysis from last year.
Yeah,
we did not check.
The geographic distributions of the species that were encountered were like remarkably similar. So if our assumptions about prevailing wind and the distribution track, then I would maybe expect that we would have encountered some similar patterns. We were going same place, the same time of year, but that would be an interesting.
You could do an honors distance that way.
Yeah.
That means we'd have to sail the cruise truck a couple more times.
I'll be okay with that.
One interesting note on that, gale number 2 at least happened around the 30-ish degree latitude, which is called the subtropical low of the force latitudes. And typically winds are very calm there, but we were in 40 knot. So our sampling there, or lack of it, was atypical.
Questions?
Like my question was just stupid.
No.
Maybe language variance in that some years you find that it's just like I think it was in 2020 but we had the Mississippi increase and we had a long time and then I mean it's just that depending on the moon phase, we have suicides on you notice that sometimes you have no change when you it's more than you know. Would you say that it's just or are they a sign of, for example, June events that is really also almost about almost all of them
of the beach. So you say that when you see NOR and when you see too much transmission, would it be that's it? Or would it be like just a true something because of friends?
So,
yeah,
oh, what's the question?
Yeah.
Oh boy. No, I'll get, and tell me if I mess something up, because I want to get that right. So the question was that here on these islands, there was in like 2019, 2020, there was a time where a lot of manowar and other jellyfish sort of showed up on the beach and in the water all at once when there was like a week-long low tide. And that there have been like jellyfish blooms or times when there's a lot of jellyfish, and is that a sign? And then one of those episodes coincided with a really big coral reef bleaching event.
Yeah, so this is happened during the rainy season, which is warm ones. And but also during winter season, which is like starting next month. And then like had cycles of like large jellyfish, and then like just this cycle of maybe a bad sign.
Yeah, so then that's also, or that's more normal during the rainy season, or?
I've noticed.
Okay.
I'm not based, it's not based on scientific.
Yeah. But living here, you observation.
Yeah.
So know a lot of things that we don't know. When we only come
Yeah,
here.
yeah. So the question is like, is it a like freak event kind of, or is it more like normalized, like with the rainy season or something like that?
I know that jellyfish blooms are becoming, where like a ton of jellyfish show up in the water, are becoming more common in other parts of the world, especially when it gets warmer. That's something that can happen. So I wouldn't be surprised if the same thing is true here, especially if you're starting to see it more in the winter when that wasn't the case before. That sounds like it could be caused by it getting warmer. Anybody else have insights to add to that?
I mean, like with the whole like jellyfish are gonna take over the world thing, like that's really dramatic. But I mean, if jellyfish are predicted and like gelatinous organisms are predicted to like increase, like the amount of them is predicted to increase with like a warmer climate, then I guess it makes sense that when like there's warmer events that there would also be like just more of them. So
Yeah,
Sarah?
maybe.
It seems like.
Oh,
yeah.
Rich.
It's very common for Bolello to have major swarms that they lash to shore.
Yeah.
Perfectly natural. So, ships of corn or ships of wind, so could be what makes sense.
Yes, it's kind of a combination of natural and induced. So things like, this is also the same for things like harmful algal blooms, right? Like harmful algal blooms have always existed. Jellyfish and salp blooms have always existed. But the level at which they bloom to, or the intensity, the abundance of them,
or the frequency,
the distribution, the size, or the size of the distribution of them, right? Like harmful algal blooms are slowly moving all the way up the coast of the United States. Similarly, if you're seeing way, more and more, the impact that they have, the distance they can reach, is an influence of overall climate change, and it's still cyclical. Within that, but it's increasing on the climate change, like bigger scale. Those cycles are getting bigger and bigger as they happen. And that has to do with what they were talking about, like these climate change winners of jellyfish, because they can tolerate these conditions better than a lot of other species.
And in terms of like the data that we've been taking year over year, we did pass through a really big bloom of Physalia that like a large portion of our data came from. There was one tow where we caught almost 30 Physalia.
27
just in that net, towing it for a mile. And I think that we encountered a similar bloom in a similar area during last year's cruise track. So obviously that's not a super strong baseline, but that'd be something that'd be really interesting to keep on monitoring, is whether that's like an area where they're always more abundant, or whether those blooms like increase in intensity.
And it is one of the weaknesses in our data that we're here the same time of year, right? Like we get here just as it starts, or as we're coming into winter. And so it would be interesting to come back to have any data from the warmer months. Because that we might find. That might be how we end up finding Physalia in the tropics.
It also might be how you end up dying in a tropical storm.
We got close. We got close.
We just welcome some.
We'll be fine. We'll be fine. Yeah, get up here.
Alright, all the groups, there's a few questions from our home. Alright, Aaron Bryant says,
switch one.
I'm so impressed. Thank you for communicating. Keep talking about how organisms respond to changing conditions and how growing industries like oil exploitation and deep sea mining will interact with environments. So important. Whom do you wish that you could tell about your research?
Oh, Aaron, you're so smart.
We talk about our science and our discoveries.
Everyone who will listen.
Yeah. Listen. Even if they don't know anything, even if they've never really heard the words like chlorophyll A or like phytophores, myctophids, copepods, like I know a lot of us coming into this didn't really know that much about zooplankton especially. And it's just, I think, taking that curiosity and being like, oh, well, what is this? And maybe finding ways to connect it with our daily lives and how it can impact us on a broader scale. Because a lot of times we're like, oh, a tiny fish in the middle of the ocean doesn't impact me, but it does. So I think really anybody, and if they're willing to listen and finding ways to make it accessible for everyone.
I'd like to add that we're really excited to have some local representation in the audience.
Heck yeah.
Our students have learned a lot about how scientists in the past have really messed up when we go to island communities and study things in these waters that are so important to local communities. And how we really need to be interacting with local communities, not only because their knowledge is so important, but also because if we're finding out things about their environment and their community and things that they're so connected to, like the Polynesian culture is so connected to these open ocean environments, and they were traversing them before we did. It's important to let them know what we found and talk to local communities about this. So our students have learned how important that is, and they're really good advocates for that. And yeah, it's also a really cool thing to be able to speak to the community.
It's not a question, but I want to say thank you for inviting me. I think it's very important to be good.
Thank you for those.
Thank you. So I would like share, I can just talk on something, but I can talk about my people around me, because I work in science. So I don't know, again, guys collaborate. So do something like when I see this, I'm like that's like studying science and environmental things. So I don't know, maybe in the future you guys can contact me about really like share to other locals, not just me. Yeah.
Yeah. All right, one last online question at least. Craig Martin says, great job, everyone. I don't want to put you on the spot, but are any of you thinking about ways to continue this same or similar research back at your home schools or maybe in graduate school?
I can speak to that a little. I'm not planning on continuing this specific project, but my project at home also has a lot to do with calcifiers. And I actually just talked to my thesis advisor. So this is going to be my senior thesis.
Wow.
Yeah. So we've talked a lot about how a lot of our critters can help take carbon into the deep ocean, but looking at carbon circulation in the ocean with currents is also really important. So my work at home is looking at a model of how carbon circulates in the ocean. And one way we can tell how accurate this model is a really cool thing is a lot of calcifiers, you can tell how much carbon was in the ocean
like years ago. So when they first started growing, because when they make their calcium carbonate shells, it records like information about carbon in the ocean. So I'm using calcifiers to check how accurate this ocean circulation model is. So yeah, I am continuing some form of calcifier work.
Yeah.
I'm also not exactly related to this research, but I will be continuing a research project back at school for my honors thesis this coming semester, but more focused on terrestrial, like freshwater aquatic ecology. But I am definitely interested in applying some of the knowledge that I have learned from this program and bringing it into that, especially with some of the techniques we've used, and even maybe comparing it to some saltwater systems. It's a bit hard because I go to school in Minnesota, so freshwater is what I've got. But I'm definitely interested. I think for me, this is the first time I've really done any like big like oceanographic research, and it's definitely piqued my interest. So it's not out of the picture forever. It'll definitely stay in the back.
Are you saying you're going to grad school?
Yeah.
What are they trying to figure
I would just note that even if individually at our own schools, we don't take specific research from this cruise and continue on with it, I think I can speak for everyone in saying that we've all been like so inspired, and this cruise track and the science we've gotten to complete and the people we've gotten to meet and everything that we've gotten to do on our grand voyage to French Polynesia and then spending time here has just been really really touching, and will continue to impact our lives and what we do in the future.
Thank you.
I have something really quickly to add just about sort of longitudinal studies, and from my perspective as the, the program director and advisor is, have to be on camera. Is that we in the past for some of the, you notice some of these projects were repeated year to year, and in particular the myctophids. We had a student last year who used it as her honors work, and for the onboard sequencing, I don't know how much the audience might know about DNA sequencing, but it's not something that is typically done on board of a sailing vessel. And Oxford Nanopore Technologies, which is the group that makes the really portable platforms that we use on board. I was a part of their education beta group, and so for the myctophids in particular, one of the reasons why we have such a large data set is because they donated some reagents to me, and I was able to work with one of last year's students, Megan Grossman, who did sort of additional sequencing. And so this is the kind of thing where building year to year and having involvement with the companies that are like producing our biotechnology, who think it's really awesome that we take these things onto a sailboat and shake them about and they still work, is a really fun facet of this particular program, but also it adds to going back year to year. And as Rick Miller has said, this is the best cruise track. And so repeating this cruise track longitudinally, in particular for the genomics, as well as the physical oceanography parameters, is really important. And you guys have said that over and over again. But yeah, the honors thesis component for all these students who go back and might want to do more work is pretty amazing to have that opportunity and be able to take those tangents and continue. Think about it.
I'm also a crier, so I can't say very much. I teared up after all their presentations. But I was also so inspired by you guys, and you're such a good group. So I just wanted to say that.
I also want to mention, I'll actually move the computer now. I want to mention that we do have, in addition to our amazing local representation, we do have some of our family and crew here in the room. But thank you all for coming. And before we all start crying, no, I'm really proud of this group, and they did some really amazing work and came through some really phenomenal sea states and a lot of personal growth, and I think they'll take this with them forever. So thank you.